Welcome to this explainer. Today, we're diving into section 2.5 of our source material, and we're going on an epic, microscopic road trip, tracing the incredible biological processes of fertilization and implantation. It's essentially a journey of survival, timing, and total transformation.
I mean, how exactly does a single, tiny, microscopic cell transform into an actual pregnancy? It sounds like it belongs in the realm of magic, right? But as we'll see today, it is deeply rooted in remarkable biology. This transformation is a sequence of highly specific chemical and physical challenges. From navigating complex anatomical pathways to executing precise genetic combinations, finding out how these biological hurdles are solved, step by step, is just truly fascinating.
So, here's our chronological roadmap for this explainer. 1. Journey to the ampulla. 2. The moment of fertilization. 3. Determining the baby's sex. 4. Cleavage and cell division. And 5. Implantation in the uterus. Let's get right into it.
Part 1. Journey to the ampulla. Our microscale road trip kicks off with insemination, which is when motile sperms are released into the female genital tract and begin their absolute sprint.
During copulation, semen is released into the vagina, and from there, these sperms have a massive distance to cover relative to their tiny size. They swim incredibly fast, passing first through the cervix, then entering into the uterus and pushing forward with one urgent, ultimate destination in mind. That destination is the ampullary region of the fallopian tube. They have to navigate this entire reproductive tract efficiently just to even have a chance at fertilization.
But here's the thing, just getting to the finish line isn't enough. Timing is literally everything here. The ovum, which is released by the ovary, is also transported to this exact same ampullary region. Fertilization can only happen if both the ovum and the sperms arrive simultaneously. It is a tiny, extremely precise window of opportunity, which completely explains why not all copulations lead to fertilization and pregnancy.
Okay, moving on to part 2, the moment of fertilization. Let's assume the timing is absolutely perfect. Let's move to the ampulla and see what happens at the exact climax of this journey, when a sperm finally meets the ovum. This brings us to the actual process of fertilization itself, the fusion of a sperm with an ovum. But the ovum has a highly advanced security system. It's surrounded by a layer called the zona pellucida. When the very first sperm comes into contact with it, it induces immediate changes in the membrane. It basically acts as a brilliant biological lock that slams shut, completely blocking the entry of any additional sperms. This ensures that only one single sperm can fertilize an ovum.
But wait, how does that winning sperm actually get inside in the first place? Well, that is where the acrosome comes in. You can think of the acrosome like a biochemical key to the ovum's lock. Its secretions help the sperm chemically dissolve and literally tunnel its way into the cytoplasm of the ovum, passing right through the zona pellucida and the plasma membrane. This is what allows the vital fusion of the male and female gametes to finally take place, resulting in a diploid zygote.
Part 3. Determining the baby's sex. Let's hit pause on our physical journey down the fallopian tube for just a second to look at the fascinating genetic math happening right at this exact moment of cellular fusion. Now, who actually determines the sex of the baby? What's really fascinating about our source material here is how this biology completely debunks a long-standing societal misconception. Historically, in a lot of cultures, women have been blamed for the sex of the baby, especially if they give birth to daughters instead of sons. But let's look at who is actually biologically responsible. It all comes down to the chromosomes. The chromosome pattern in a human female is XX, while in a male it is XY. So all the haploid gametes, the ova produced by the female, carry the exact same sex chromosome, an X, always an X. However, the male gametes, the sperms, are split down the middle. 50% carry an X chromosome and the other 50% carry a Y. Because the mother can only provide an X, it is entirely dependent on whether the father's fertilizing sperm carries an X for a female baby or a Y for a male baby. So scientifically speaking, it is correct to say that the sex of the baby is determined by the father and definitely not by the mother.
Okay, part 4. Cleavage and cell division. With the genetics locked in and the zygote farmed, we are back on the road. The zygote doesn't just sit there in the ampulla. It immediately gets right to work and begins to move down the isthmus toward the uterus.
And as it travels, it undergoes rapid mitotic cell division in a process called cleavage. Think of it like an exponential growth montage. That single cell divides into 2, then 4, 8, and 16 daughter cells, which are known as blastomeres.
Once the embryo hits that 8 to 16 blastomere stage, it's called a morula. But it doesn't stop there. The morula just keeps furiously dividing and transforms into what's known as a blastocyst as it moves closer and closer into the uterus.
As this blastocyst approaches its final destination, its cells undergo this incredible level of organization, arranging themselves into two very distinct parts. The outer layer of cells forms a ring called the trophoblast. Meanwhile, an inner group of cells physically attaches to the inside of that trophoblast layer, and this is called the inner cell mass. This distinction is absolutely crucial because these two parts have drastically different jobs coming up. The trophoblast is going to handle the attachment, while the inner cell mass will eventually differentiate to become the embryo itself.
And finally, Part 5. Implantation in the uterus. We have officially reached the end of our microscale road trip.
The rapidly dividing blastocyst has completed its journey through the oviduct and arrived right in the uterus. This is the macro-scale anatomical destination, the specific environment where this microscopic bundle of cells will secure itself, eventually developing into a fully formed fetus over the next 9 months.
So how does it actually secure itself? Well, remember that outer layer we talked about, the trophoblast? It gets attached to the endometrium, which is the inner glandular layer lining the uterine cavity. Once it physically latches on, the uterine cells kick into high gear. They divide rapidly and literally grow right over the blastocyst to completely cover it up. As a result, the blastocyst becomes fully embedded within the endometrium of the uterus. This embedding is the literal biological definition of implantation, and it officially marks the start of a pregnancy.
We've witnessed an amazing biological ballet here, full of survival, perfect timing, and rapid cellular organization. But it leaves us with an incredible question. Just how does this newly embedded tiny inner cell mass know exactly how to differentiate, layer by layer, to form the complex tissues, organs, and beating heart of a fully developed human being? Well, that is a whole other explainer. Thank you so much for joining me on this micro-scale road trip through the very beginnings of human reproduction.